So Casey is a second-year graduate student in the physics department here at UCLA. Casey is broadly interested in many topics, including evolutionary biology, and recently he's trying to apply this to immune system. Casey was studying, I think, both chemical and biomolecular engineering and physics at University of Pennsylvania before he joined us 2 years ago. So today, I think Casey will tell us about the behavior of populations when they both move and evolve in heterogeneous space. So.
Okay, yeah. Thanks for the introduction, and thanks to Shanshan for her guidance with the project. Environmental heterogeneity, meaning spatial variation in an environment, is ubiquitous in nature. And this picture on the left shows an especially extreme example of this, where there's desert, which meets some lush greenery, which means a river, and they're all in contact with the air. And of course, there are organisms that live and evolve in all of these environments. And a question to ask is, how much does the heterogeneity of an environment affect the evolution of the organisms that live there? And I think it's clear that the answer to this depends on the migration rates of those organisms, meaning the rates at which those organisms explore their heterogeneous environment. So if you imagine a frog, which might go back and forth between the bushes and the water, it will face selection pressure from both of those environments and will need to be well adapted to both. But a fish in the river doesn't face any direct interaction with the desert, although it would interact with other organisms, which might interact with the desert. So to address this question, we study a model of one-way migration between two distinct habitats, with the goal of understanding how the steady state populations in the model and the dynamics near steady state depend on migration rate. So just starting with a brief overview of the model, we have two habitats. So habitat 1 is here and habitat 2. And these habitats are populated by a population of asexual organisms, each with some genotype specified by a number between 1 and m. And organisms migrate from habitat 1 to habitat 2 at some rate k. And the migration is just one way, so from 1 to 2, but not back from 2 to 1. Then mutations occur at a rate gamma, and these two habitats are distinct in that they each exert different selection pressure specified by a fitness landscape for each. And then there's also a parameter rho, which specifies the degree of resource limitation in each habitat. So the bigger rho is, the more severe the resource limitation. And it might seem rather specific to focus just on one-way migration, meaning just from 1 to 2, but we have two motivations for this. The first is that there are real-world systems which have a mechanism that limits migration to one direction. So one example is gut microbiota, which are carried by the flow of food through the GI tract. And another example is are organisms that live in rivers and streams that are carried by the current. And we're not experts in either of these areas. And so if people in the audience have insight into the applicability of our model to either of these or to other systems, we're interested to hear. And the second motivation, which was actually our original motivation, is that simple models often give insight into more sophisticated models. And in fact, we've shown that that's the case here. So the results that I'm going to present today, we've shown separately, do generalize to a more complicated model where there's networks of habitats. So before specifying the model mathematically, I just want to give a brief description of how the populations behave. So typically the habitats support distinct quasi-species. And this picture just demonstrates what I mean by quasi-species. So here on the x-axis, we have genotype. In this model, we just have 34 different genotypes. And the blue curve shows the fitness of each genotype. So here, this is the region where fitness is the highest. And as you would expect, there's a locus of population around this region of genotype space. And that's what we call the native or the well-adapted quasi-species. But now, in this case, there's also migration into the habitat. And due to the migrants entering the habitat, there's this other locus of population over here. And that's what we call the non-native quasi-species. So there's this coexistence between the two quasi-species. And as we'll see, there are several critical migration rates at which one or the other of these quasi-species goes extinct. And when this happens, the dynamics exhibit critical slowing down. And that just means that the relaxation time of the system, meaning the time at which it relaxes to steady state, becomes very long. So the dynamics are very slow. And it's actually known in the ecology literature that critical slowing down is often associated with extinction. But what's interesting about this case is that we didn't start out with an ecological model where there's distinct species. We just start out with one species, which can take on a variety of genotypes. But from within that model, there's the emergence of quasi-species, which then interact and give rise to this critical slowing down, which is known to occur in ecology. So moving on to the mathematical model, so let's define these variables. So n sub 1i, this is the number of organisms in habitat 1 with genotype I, and similarly in habitat 2. And it's convenient to collect these numbers into vectors. So here the vector n1, the ith component would just be the number of organisms with genotype I. And these populations obey these two coupled differential equations. So the first one is for habitat 1, and then the second one is for habitat 2. Going through term by term, this first term is just a matrix V, which acts on this on the vector of populations. And the matrix describes fitness-dependent reproduction and mutation. And the particular definition of V is here. And you can see that here it's phi, that's the fitness landscape. So that describes fitness-dependent reproduction. And then here gamma is the mutation rate, and this term describes mutation. The second term is pretty much a standard logistic growth term, which discounts the growth rate in proportion to the total population. So N2 tot, that's the total population in habitat 2, and similarly here is the total population in habitat 1. And so this discount to the total growth rate is scaled by this parameter rho, which describes the severity of resource limitations. And then finally, there's migration. So we're subtracting this term from habitat 1 due to the migrants leaving habitat 1, and then that gets added to habitat 2 because the migrants enter habitat 2. One thing to point out is that here these populations are treated as continuous variables, whereas of course in real life they should just be whole numbers. But this is a good approximation to treat them as continuous variables for large populations. And this model that I've described here is a slightly modified version of a model studied in this paper by Wang, and our analysis was largely inspired by their approach. So there's a trick for understanding the dynamics, which is helpful, but it's not essential. So if you don't want to pay attention to the math here, you'll still be able to understand what comes next. But recalling that this matrix V describes fitness-dependent reproduction and mutation, we can look at its eigenvalues and the eigenvectors, and we'll arrange the eigenvalues in descending order. So lambda superscript 1 is the biggest. And these eigenvalues have a particular interpretation, which is that they give the intrinsic growth rate of each eigenvector. And that means it's the growth rate of the eigenvector in the absence of migration and in the absence of the resource constraint. So the intuitive picture you can have is that these eigenvectors compete with one another in the habitat, and their competitiveness is determined by their intrinsic growth rate. And as time goes on, the psi superscript 1, the eigenvector with the greatest growth rate, just outcompetes all the other eigenvectors. And so finally, we're just left with the steady state population at the isolated. So ISO stands for the steady state population in the isolated habitat is just proportional to this eigenvector with the largest growth rate. And a key takeaway from this is that this eigenvector is localized around the most fit region of genotype space. And accordingly, the population is mostly made up of organisms of high fitness genotype. So now looking specifically at those populations, so the steady state populations with 0 migration rate. So this just means the two habitats are isolated. Again, the blue curve is the fitness landscape. So here where the fitness is high, here's a place where fitness is high, and then down over here it's low. And as it turns out, it's a little bit complicated, but it turns out that here where this region of high fitness is the widest, that's the most fit region. And because of that, this is where the population is localized. So all the population is localized around the most fit region. And here's the formula. And as you can see, indeed, the population is proportional to that most highest growth rate eigenvector with some proportionality constant. And it's also can be seen from this if you just add up all the components. So that just means adding up all the populations of all the different genotypes to get the total population. That gives you the carrying capacity. and it's proportional to the maximum growth rate, which is intuitive, right? The higher the growth rate, the bigger the carrying capacity, and it's inversely proportional to rho, which is the resource limitation. So the more severely limited resources, the lower the carrying capacity. And the same picture applies just as well to habitat 2, except for here, the most fit region of genotype is in a different place in genotype space. So one thing to note is that we've used what I'm calling a linear genotype space, meaning if you look at some genotype, let's say genotype 30, it can mutate to genotype 31 or to genotype 29, but it can't mutate all the way over to, let's say, genotype 20. So of course, this isn't the way that real genotypes work, but actually all of our formulas are totally general for any choice of which genotypes can mutate to which others. So these formulas are perfectly valid for the more realistic genotype space. But for the purpose of visualization, it's easier to just use this linear genotype space. And as it turns out, there are two important possibilities to consider here. The first possibility is that these populations, when k equals 0, are non-overlapping in genotype space. And that's what you see here. Here, this population has genotypes around 22 to 30 or to 34, and this has genotypes 2 to 10. And so they're non-overlapping. They're in separate parts of genotype space. And this is the case where the two populations behave as distinct quasi-species, and where critical slowing down accompanies the extinction of either quasi-species. The other cases where they do overlap in genotype space, and that's just the less interesting case. So we'll focus on the first possibility. So now that we've talked about the populations for no migration, now we can actually answer the question, what happens when there is migration? So in habitat 1, in a sense, not much happens. The shape of the population, meaning the distribution of genotypes, stays the same. All that's happened is that the population is lower because there's all the migrants leaving, and that reduces the population. And this formula for the steady state population is exactly the same formula you saw on the previous slide, except now we're subtracting k. So that just means there's this discount to the population due to the exiting migrant organisms. And you can see if you make k big enough, like if you make k all the way up to lambda 1, 1, then the whole thing is just 0 because these just cancel. So that means if you make the migration rate big enough, the habitat just becomes empty. And if you make k even further, it just remains empty. So there's some maximum migration rate that the habitat can actually support any population. So now looking at considering the fact that these organisms are going to migrate from habitat 1 to habitat 2, we can look at essentially the total number of organisms that are migrating. And so that's quantified by the migrant flux R. So here on the x-axis, we have migration rate, and on the y-axis, we have this migrant flux, which is essentially the number of organisms that are migrating. So when k is 0, as you would expect, there's 0 migrant flux because there's no migration. And as you increase k, then the migrant flux increases. But if you increase k far enough, then the depletion of population due to this discount, due to the exiting migrants, the depletion of population in habitat 1 causes the flux to actually decrease as k is increased too far. And so as we'll see in just a second, there are these 3 particular migration rates at which one or the other quasi-species goes extinct. And at these migration rates, that's where critical slowing down occurs. So moving from habitat 1 to habitat 2, starting at k equals 0, so that's with no migration, the population is just localized around the most fit region, as we discussed. But now if you have positive migration, then this native quasi-species, the well-adapted quasi-species, competes with a non-native quasi-species, which is being fed by migration from habitat 1. So there's this coexistence of these two quasi-species. And if you increase k far enough, the non-native quasi-species here basically beats out the native quasi-species. And so that's what's shown here. The native quasi-species has gone extinct, and we're just left with non-native. And that describes what happens when you increase k from 0 up to k minus being the point where the native quasi-species goes extinct. And you can sort of see that if you increase k even further, you just expect the same thing to happen as in reverse as we come down the other side of this curve. And that is indeed what happens. So at k plus, the population habitat 2 here looks the same as it did at k minus. And as we increase k further, you get a reemergence of the coexistence between the two quasi-species. And eventually the other quasi-species, the one native to habitat 1, goes extinct. So this can be summarized in a few plots. This is the plot for the flux we were just looking at, but here summarizing the total populations of the different quasi-species. So on the x-axis is migration rate. For zero migration rate, this orange curve, which is the population of quasi-species 2, the one native to the set to habitat 2, the entire population is just the native quasi-species. But when k is increased, that native quasi-species begins to coexist with the non-native quasi-species, indicated in teal, due to the migrating population. And eventually, for high enough k, the native quasi-species goes extinct because it's replaced entirely by the non-native quasi-species. And then again, if you increase k even further, the same thing happens in reverse. One interesting thing to note from this plot is that if you just look at the total population of the habitat, which is shown in this dotted gray line, that total population is constant below k minus, so before extinction occurs, for migration lower than when it occurs, but then it jumps up and has a positive slope at right where after extinction occurs. And so a possible implication of this is that it provides a method for experimentally determining where this extinction event occurs without requiring the need to actually do DNA sequencing to determine which particular quasi-species are existing. Another thing that's interesting to look at is the genetic diversity, and the importance of genetic diversity would depend on the particular application. But for example, in the case of the gut microbiota, having diverse gut microbiota may be very important for health. And as is fairly intuitive, the genetic diversity is maximized at this region of intermediate K, which corresponds to this region over here, where there's the coexistence of the two quasi-species, right? It makes sense. If there's two quasi-species coexisting, the diversity will be higher than if there's just one or the other quasi-species. And finally, we can look at relaxation time, which quantifies the time scale on which the system relaxes into steady state. And as you can see, the relaxation time has these peaks at each of the 3 critical migration rates at which extinction occurs. And this indicates the critical slowdown associated with extinction. So defining the relaxation time tau more precisely, if you imagine perturbing the system from steady state, then you can look at the deviation of the system from steady state and how that deviation changes in time. And so that's what delta n here signifies. That's the deviation from steady state. And for some genotypes, that deviation will decay really quickly, but it turns out that there's a particular distribution of genotypes which decays much more slowly than the others. And so that's what this zeta vector is. That's the distribution of genotypes, which is very slow to decay. And this equation just tells you how it decays. So it decays exponentially with some time constant tau. And then there are other terms, but they decay much faster, exponentially faster than the first term. And this is just mathematically defining what zeta is. But the important thing is this relaxation time, that's the time constant for this slow distribution to decay. So what happens is that at the phase transitions, this means that at the critical migration rates at which extinction occurs, tau, this time scale for decay, goes to infinity, meaning the decay is very slow. And in this case, zeta decays sub-exponentially slower than any exponential function. And in fact, in particular, it decays as 1 over time. So looking in particular at this distribution, the slow decaying distribution, is helpful for getting insight into the actual mechanism of what's happening. So the distribution corresponds to a redistribution of population from one quasi-species to the other. So for example, here at this distribution at K minus or K plus, this is where quasi-species 2 goes extinct. You can see that when it corresponds to a decrease in the population of one quasi-species and an increase here in the other, or vice versa. It could be an increase in this quasi-species and a decrease in this other one. And the same intuition applies over at this other critical migration rate where the quasi-species 1 goes extinct. The if there's a decrease in the population of this quasi-species, there's a corresponding increase in the population of the other, or vice versa. And it turns out we can actually quantify the exact dynamics of this deviation from steady state, which is proportional to this to zeta, and it obeys this equation. And at first glance, this equation might not seem very interesting, but the key point is that the only term over here has this rho in it. The rho was from the resource limitation. And so the observation is that all the terms in the dynamical equation from before, remember there was the term with the matrix V describing fitness-dependent reproduction and mutation, and then there was the migration term. All of those terms have canceled, and the only thing we're left with is the term due to the resource limitation. And so that's what it says here. We're just left with just the resource limitation. And the interpretation of this is that this slowly decaying deviation zeta isolates the ecological process of resource competition between the two quasi-species. It isolates that from the other evolutionary processes in the model. And furthermore, as one quasi-species goes extinct, the resource competition between quasi-species, which is proportional to the two quasi-species population, vanishes, right? As the population is vanishing, then the competition vanishes. But that competition is the only restoring force bringing the system to steady state. That's what we saw here. The only restoring force is due to that competition. So if the restoring force vanishes as the system approaches steady state, then there's essentially nothing pulling the system to steady state. And we get a really slow dynamics. And that's the cause of the critical slowdown. So just to summarize all of our results, the migration rate determines which quasi-species exist at steady state. And there are a few particular migration rates at which one or the other quasi-species goes extinct. Quasi-species extinction causes critical slowing down. And this is an example of critical slowing down, which is associated with extinction and has been studied in ecology. But in our model, we don't start out with an explicitly ecological model with distinct species. We just have one evolutionary model, and this ecological process emerges from within it. And so we think this model may be relevant to the evolution of gut microbiota and possibly organisms in rivers and streams. And like I said, I'm interested in hearing any ideas along those lines or for other systems. And finally, the key intuition that I've discussed about the competition between quasi-species and critical slowdown does generalize to more sophisticated and more realistic models where there's many habitats and two-way migration between them. So that concludes my talk, and I'm happy to answer any questions. Thank you.